Yo, and welcome to Shooting the Soil. Alright, so today we are going to be checking out our outdoor raised bed. We just had to till it up because it got infested by roots from the forest that's right next door. So gonna have to find a long-term solution, maybe dig a trench around it or something. But anyway, so we threw down some mulch right on top of it and planted some cover crop. We did a fenugreek and a hard winter wheat. Yeah, so wanted to do a benchmark at the end of the season and share some microscopy with all you guys. And yeah, let's see how this outdoor bed is looking as far as the biology. Yeah, we're in southern Massachusetts, by the way. We haven't quite had a freeze yet, but we've had a couple of days in the or a couple of nights, I should say, in the mid and high 30s Fahrenheit. Yep. So, see a nice fungal strand there. We're up at the top. Anyway, other than that, main thing I want to talk about is nutrient cycling. So the main benefit of planting a cover crop is they're going to harness that energy from the sun, and through photosynthesis they can pump 80% of their energy down into the soil through exudates, feeding the biology and making the whole system go. Yeah, here actually I had an image here that I just made up, so you can see how it's coming from the sun and then cycling through the soil of the stomach with all the microbes, the fungi, and earthworms and bugs breaking things down, decomposing, doing their thing. So we want to take advantage of that extra sun energy. And the winter wheat, last couple of years I've grown it, pretty much stayed green through all of winter. We got a nice, that might be a root right there. Oh yeah, we're at 100x magnification right now, just kind of scanning through. And hi Jay Dixon, thanks for joining. Glad you can make it. Alright, so we're going to scan around here. When we go into 400x, we'll be able to see some bacteria. Right now, I'm just kind of scanning through. We want to see, judging the amount of aggregates that we have here. Let me know if you guys see my mouse. Sometimes it turns my mouse capture off, but I'm pretty sure it's on right now. Anyway, so we want to see the aggregate level. Dilution, if anybody wants to know, is 1 to 10. So that's 10 parts water, 1 milliliter of soil. Then we mix it up in the tube. And we put 2 drops on the slide. Alright, seeing a couple of fungal hyphae. Some small ones. Not too shabby. Let's just scan around here. Move my chair up a little bit. And yeah, how's the audio and everything? I got a new mic boom here, so the whirling sound of my computer fans wasn't bothering anybody. Hopefully it sounds much better. Alright, yeah, I got some nice fungal hyphae. Nice little small one there, going through, grouping up that aggregate. Oh, yeah, I do have a video here. Let me actually throw on a quick shot so you can actually see the bed of what it looks like. Back over to the microscope. And that was the bed. But yeah, pretty happy. We got that cover established. And yeah, depending on your situation, winter wheat isn't too hard to terminate. On a small garden like mine, I'm actually just going to go right in with the Hori Hori knife, and it's going to provide a lot of nice mulch. So I'm going to terminate just by cutting it right at the roots, leaving all the roots in there, and then planting come spring. And all winter long, it's going to be doing the best it can in its conditions to cycle nutrients. Pump that liquid sunshine down into the soil. Feed those microbes the cakes and cookies, like Dr. Elaine says. All right, yeah. So we're seeing a lot more nice large aggregates over here. Oh, still scanning up and down. Be surprised if we see, since it is so cold, if there are any nematodes and whatnot. They might be. But it was decently warm. I took this sample yesterday before it got cold to try to hopefully preserve some large microorganisms. But you never know what you might see. We still might see a nematode or a microarthropod, maybe a rotifer, but it is pretty late in the season. But we've had a lot of days. It's been surprising. In the 70s, early November and late October. For New England, that's pretty warm this time of year. Usually we have our first snow already. Ah. Thanks, Craps Dealer, for letting me know it sounds good. Nice to see you here. All right. Yeah, a whole bunch of aggregates as we're getting towards... we're only about a quarter of the way through our scan here. But let's actually jump into 400x just so we can see some kind of life in this soil. We'll see how, and take a look at how the bacteria is looking. Oh, let me back that off a little bit so I don't bump into the slide. Let me get dialed here. Oh, am I going the wrong way? I think I am. Yep, a little bit closer. Let's get some more light on the situation. There we go. Focus. 1 second. Alright, I'm going to use the big one. It's always tough because you don't want to bump into the slide because everything starts moving around on there. Very close. Alright, there we are. Now we can use the fine focus. Let's adjust our light a little bit more, brighten things up. Alright. So now we can see a bunch of bacteria wiggling around in there. See if I can get a little bit more definition. Yeah, there we go. See what we got for shade. Alright, so not super high counts of bacteria, but not surprised at this time of year. And we just did stir them all up when we had to get all those roots out, breaking up their home a little bit, but they'll get reestablished. Yeah, decent amount. Got a couple of the different shapes and sizes. We got the rods, the bacillus shape, the round one, the cocci, another rod over here. All right. So yeah, feel free to ask any questions in the chat of what we're looking at as far as gardening and microscopy goes. Over here we can see a little fungal spore tucked against that little piece of sand, or actually that's pretty small, so it'd be silt. Just a little mineral. Nice big aggregates. And see, small little hyphae there. Pretty clear. Most of the hyphae that we see in the scope are saprophytes, the reducers, just breaking down that organic matter, weaving in and out, helping form that soil sponge, holding things together. Oh, we got little football, another fungal spore right there. See quite a few of those. All right. Some little tail right there. Could be a small fungal spore. Kind of looks like that. Just starting to sprout. Might not be the best time, buddy. But better than never. Alright. Got all sorts of hyphae. Another nice big saprophytic looking fungal hyphae there. Yeah, basically I just took a broad fork and went through and ripped out all the roots that came into the garden, because they were really matting up my soil and giving what I was planted in there really no room to grow. So we did as minimal as possible and then covered it right up with some mulch and planted that cover crop right into it. Try to reestablish and get things functioning over the winter so it's ready to go come next spring. Then I'll have a ton of mulch with that winter wheat. It'll probably get 2 to 3 feet high by the time I'm ready to plant come our last frost. Yes, they would overall in this bed my aggregation is improving. Even though I tilled, probably broke up a lot of these big pieces, but still seeing a lot. But just everything getting that nice brown color. That's a nice septi there in that fungal hyphae. But see, even this clear mineral is slowly getting those aminos out. Amino acids, turning it brown as bacteria and other biology goes to town, mines nutrients from it, and excretes that glomulin, different acids to do the pH and extract different stuff, which will eventually be reduced and transferred to the plant. And then recycled all over again. But, as far as I might have read the question wrong, I try to be as no-till as possible. This is the first time I've tilled this bed in, I think, I would say 4, maybe 5 years. And it definitely didn't need the aeration. It was fluffy as can be. But, man, was there a lot of roots in there. I'm going to be coming out with a video on the tilling project and just how I did it and whatnot. And yeah, you'll see just handfuls of roots of just a small area. Yep, so if you are planning on building a raised bed, I would definitely not build it 2 feet from, or probably about 4 feet in my situation, from very established forest. The big maple tree, and I had a invasive bittersweet vine that I've been trying to eradicate out of there. That grew into the garden. Yeah, I would. Well, when they're alive and they're deciduous and they don't break down, that's the problem. I would have loved to leave them in there, but the other option was if I rented a trench digger, I might have been able to kill the roots by doing a trench around the garden. That was my other option and might actually be my final solution that I have to do. But decided just to rimp about instead of renting the trench digger. What do we got here? There's a nematode. Very still nematode, just his tail. His head. Pretty sure that's a nematode. Yeah, he's not moving much. A little too cold, probably already hibernating. Oh, he's moving a little bit. And just got his head wedged into that little aggregate. Looks like some organic matter with some minerals all clumped in there. Yeah. So we got him in there. And then, of course, once I do the trench, how do I fill it? Do I pile stone and concrete in there? The root's going to go under it. But should help the situation. All right. Well, let's see. Yep. There we go. Here's some more hyphae. Interesting one. Got some green glow in there. But we see septi. Little separations in the cell. It's always interesting when they got all this stuff kind of globbed up onto them. But I think they're just feeding off it, breaking it down, and reducing it. This shape is interesting there, but I was trying to see if it was maybe a microorganism, but no, I think it's just a mineral. But yeah, nice big aggregation with a whole bunch of fungal hyphae. Mainly saprophytes. One thing I really want to get epifluorescence so we can really start looking at the and identifying AMF, arbuscular mycorrhizal fungi, and other mycorrhizals, but they're pretty small from what I've seen. Yeah, most of I've taken Matt Powers' class, so if you check him out and look at some of his shots with the epifluorescent scope of mycorrhizal. But yeah, so most of what we see and we've looked at today are all saprophytic fungi. Basically, they don't, they're not endophytes. They don't go into the plant, but they're good at reducing organic matter and providing nutrients. We got this looks like, I believe this is a cyst. There's an organism in there, and he or she has formed a little cocoon and gonna hold out the winter in there. It's a very interesting kind of star-shaped little cyst. Not too familiar with the different shapes and patterns and whatnot, but if anybody's seen or knows what organism might be living in there, very symmetrical. I don't think it's an amoeba, but somebody has made a little cocoon for the winter there. Alright, where are we on the slide here? Yeah, we could go over just a little bit more. We'll do one more scan down here at 400x. Yeah, I would highly recommend Matt's classes. His books are excellent. He's definitely, I love his teaching style. I've always been an audio, video sort of learner, and exactly what I exactly how I want to learn. And he does it better than most, for sure. All right. See what else we can find. Nice little clumps of organic matter and whatnot. Hmm. Yeah, I reference regenerative soil all the time. Ratios for all the amendments. Yeah, we got a fast moving little bacteria there. Or that might be a flagellate. Probably has just one little cilia. Whipping around, moving himself. Still got a bit of motion down there. Yeah, it's been seeing more and more flagellates around in my soil this year with my microscopy work. It's definitely one thing I wanted to improve on to get that balanced soil food web. All right, where are we? Yeah, we can still go down a little bit more, see what else we see. Oh, what do we got over here? That's interesting. Oh, we got a couple of amoeba. Oh, I think we got a little amoeba family out over here. All probably all clumped up together, or clumped up together. I mean, sharing heat, huh? And doing their thing, splitting and forming new amoeba. Very cool. Yeah, definitely. Look how look how wide their cell walls are. Definitely protecting themselves from the cold. Yep. There we go. Pretty nice look at an amoeba family. I actually kind of want to. I'm gonna snap a picture of that amoeba family. All right. Let's see what else we can find. Anything else cool to look at here? More clear strands. That one's a little weird looking, and I don't see any septi. Could potentially not be a good one, but since we only see one, it's not really a problem. We saw a whole bunch of other fungi that are going to outcompete, and just seeing one or two like that isn't a cause for much of a concern. If you're going to have a fungal pathogen issue, you're going to see a lot of strands like that in the soil. Yep. Another good one. See a septi. Been seeing a lot of these big ones. They might even actually be a fruiting body mushroom. With all the different stuff stuck to the hyphae that they're probably digesting. Might even be some endophytes. There are bacteria that are endophytic and go inside and live inside a fungi too. As well as plant roots. All right. Where are we on the slide? We're about halfway down. Let's scan down here. Still at 400x magnification. Seeing what else we might find. All right. Take a little zoom in on this. So here we go. We got some organic matter. Probably a piece of a leaf breaking down. There might even be a fungal hyphae right here. It actually looks like it growing on right along the outside of it. Let's see what we see over here. Yeah. That's what organic matter looks like under the scope. And another fungal hyphae. Nice clear septa, very uniform. Another good reducer. Yeah, everything nice. Forming nice clumps. Soil structure is going to be back in no time. Another fungal hyphae, right? I mean, not a hyphae, spore. Fungal spore right there. Actually, sometimes assisted organism, like a little tiny protozoa or a flagellate, can actually be mistaken for this because it's got that outer cell wall. Looks a lot like a spore. Colored a lot like a spore, but cysts can actually look a lot like that as well. So it could be one or the other. Most likely a spore, though, because we see a lot that look like that. All right. Yeah. So our fungal levels are pretty good. I wouldn't say we're quite, still a little off the 1-to-1 fungi to bacteria ratio. That's a big piece of fungal hyphae, I think. It's just broken off right there. By the color, anyway. Alright. And we can go down a little bit more. Yep. Can always use more fungi. So this little clear strand over here is getting down to the size of an endophytic, one that would go inside a plant, like mycorrhizal. They tend to be pretty small from what I've seen, but really need the epifluorescence to positively identify them. I don't know what that is. Maybe algae or microplastic. Does have quite the glow to it. That actually might even be on the top of the slide. I just got a little piece of hair or microplastic on the top of the slide. Alright. And almost down to the edge here. Is that anything? Nope. Just a piece of silt. Yep. Yep. Another example of a little piece of organic matter. So definitely happy with the organic matter to soil ratio. I think we got a shaped like a, not seeing any internals, but kind of shaped like a testate amoeba. Little balloon shape. Alright, where are we? Just about to the end of the slide. Another fungal hyphae over there. Another small clear hyphae. Oh, there's a nice, rich, wide, very uniform piece of a fungal hyphae. This is a piece of a critter right here. And looks to me like it was a rotifor, if I was guessing, but I think he's missing. He's missing his head, from what it looks like. Some predator. Maybe an arthropod or something like that. That's how he met his end, by the looks of it. Oh, there we go. Another very interesting looking little football fungal spore. And yeah, I think that'll just about wrap things up. Anybody have any questions or things they want to say in the chat? Or, and if you're watching this later, please, happy to have a conversation in the comments. But yeah, not too bad. Overall, just a nice benchmark before the end of the season that I can go back on and judge my progress. Let's see. So yeah, this looks like a root hair, or not even quite a hair, but just the root. Always interesting how different plants kind of weave their cells together differently. All right. Oops. More organic matter. Another clear fungal hyphae, but all decently uniform. That's a long one. Wow. All the way up into here. Yep. That's definitely one of those saprophytes. Probably one of, like, when you have I did throw a bunch of coffee grinds on there, but if you leave your compost in the bucket for too long, like your food waste in your compost kitchen bin, you get those white hairs. That's what they look like under the microscope. That really long one. This. Yep. Not very many septi, really long, and fast growing. But, yep. Very fast reducer. It's another type of saprophytic fungi. See what we see over here. Yeah, but I think that's about it. Right at the bottom of the slide. Oh, I think we got one more amoeba. Amoeba's always a good sign. Nice facultative microbe, meaning they can live with oxygen or without oxygen. They can kind of survive in both, aerobic or anaerobic. So yeah, they're a good universal microbe. Got a couple of more fast swimmers down there. It's kind of hard to see, but yep, there it is. Probably another little flagellate. Or some bacteria have cilia and can swim around. There's another very little one. They can move decently fast, but most of them are protozoa that are moving quick, whipping their tails. Then the big ciliates have cilia all over their bodies, and they can get going really quick. But yeah, be surprised if we saw one of those this time of year. I usually only find them in the compost bin. But yeah. And occasionally one in the soil. But they like things more lacking oxygen, the big ciliates. Like that, the anaerobic. So they can swim around with all those little whips, all those little cilia. All right. Any last questions? I think we're just about finished. But yeah, definitely hit those buttons if you would be so kind. Share the video with anybody interested in microscopy and looking at the health of our soil in a whole new way. Just like we judge the health of our plants by looking at them. This is just another tool that we can check things out and get signs of our garden health. Yeah. I think this is a mineral. I almost thought that was a big amoeba, but not really seeing the cell wall. And I think it's just a, yep, just a round square piece of sand there. Anyway, yeah, Hans, Ketchup, got a question? Shoot. Be happy to give it a shot. Oh, look. There's another cyst, or possibly an amoeba. Finding a couple more things down in there. Definitely not seconds. No, they can live pretty good amount of time. I'm not sure the specifics, but I've had, you can have a culture of bacteria survive, especially if you put them in the fridge. Like if you have a kombucha and stick it in the fridge, you could take it out months later and still see a bunch of bacteria. Maybe some died off and are new, but I'm not sure exactly how long they could live, but I know it's at least days, if not longer. And some I think can live and hibernate and pop back to life even after an entire winter. But there's definitely probably some bacteria. There's millions of types of bacteria, mind you, too. And some may have very short lifespans, but like lactose bacillus that you use in ferments, I'm pretty sure can have a pretty long lifespan. But I'm not sure on exactly how long. But I would guess, upwards of a year, if not more. But anyway, I think that wraps it up for this stream. Yeah. We'll leave with another clear, weird looking little fungal strand there. And yeah, we will. I'm gonna work on getting that tilling video out. Show just how we're gonna go over some of the sciences, like things like the Fenton reaction and the pros and cons of tilling. And yeah, should be pretty interesting. So definitely check that out. Still gonna hopefully do a couple of lives because we do have an indoor, climate controlled small little grow room. And yeah, so and Hans has one more question. Yeah, as far as reproduction, they're pretty much just wiggling around, bumping into each other, sharing their deep DNA, splitting, doing all sorts of stuff, pretty much 24/7. And yeah, it is amazing. Like in a compost tea, you can go from billions to trillions in just a couple of days. It's pretty amazing. But all right. Well, anyway, I think that's a wrap, folks. So yeah, until next time, we'll catch you on the flip side. Have a good one, everyone.